Hi, everybody. Okay, well, welcome back, gang. Just taking a few seconds to get everybody logged in here. I hope you had a nice weekend and continue to remain healthy. Right. Okay, we'll get started here. Maybe another few people will join us in a second. Okay, so if you haven't heard the news, but I suspect you have, hey folks, we're going back to school next week. Ho ho, we're going back to school next week. So HAC has made the decision that a variety of classes, particularly those that are science classes that have labs, that we can get back on campus next week. So starting next Monday, the 31st of January, and for you here in a Tuesday class, February 1st, Tuesday, February 1st, we will be having class on the Lancaster campus as though life is actually normal. Okay, so I do want to encourage you to start thinking about what that would mean for you in terms of your personal drive to campus. That we're not used to thinking about these things anymore because it's been a while since we've had to do it. But we'll be back together on Tuesday, and as you know from your syllabus, the room we will be in for lecture is in the East Building, room 229. Room 229 this coming Tuesday. So a week from today, I will meet you on the Lancaster campus, East Building, room 229 for our first in-face person-to-person lecture. So, to me, that's very good news because we've been out of this teaching environment for nearly 2 years now, and it just continues to be a struggle to teach remotely. So, okay, do you have any questions about that news you'd like to ask? Okay, everybody's quiet, so hopefully everybody's okay with that. So don't forget your masks. Of course, we do need to wear masks while we're on campus. But other than that, things hopefully will start to get back to normal. So let's keep our fingers crossed. Okay, we're going to start rocking and rolling in chapter 2, folks. Okay, so before I do that, let me just remind you of a couple things. Okay, so the lab that you did last Thursday with the introduction to Mathematica, the first 3 chapters of the elementary introduction to the Wolfram language notebooks, they are due today, right? Today is Tuesday the 25th. They are due today. So obviously, as you complete them, you share them with me in Mathematica, and you give me editing permission, please. I will be starting the process of looking at them tomorrow sometime, okay? But they are due today. Finally, we'll also mention that your first official homework assignments of the semester will be due this this Thursday, okay? And so that includes the Mastering Genetics problems from chapter 1, questions 1 through 17. And then also the 4th chapter of Mathematica programming is due on Thursday as well. So let me go in to Mastering Genetics and show you what I'm talking about, just in case there's any mystery here. Okay. Okay. So yes, my username's Alanator. My textbook representative thought that that would be funny, so he put me in as Alanator. Okay. Okay. Okay, so once again, part of your homework that's due on Thursday is to complete the 4th chapter of the Mathematica EIWL text. Okay, and so that's okay, Terry, that's okay. I see your comment. That's quite okay. Understand technology issues. Okay, no problem, Terrell. Okay. So what you do is go to document sharing, right? And up here is Mathematica notebooks for homework. Mathematica notebooks for homework. Okay. You click open to that. You find the 4th chapter notebook, which should be here. Am I missing it? Oh, right here it is, down at the bottom, EIWL4. You download this to your computer, you upload it to Mathematica as we did on Thursday, and you complete the work that is described in the file. Okay, so that is also due this Thursday. All right, so do you have any questions about any of that stuff that is coming due? Okay, while I have you here as a captive audience, this Thursday, we'll still be doing a remote lab to the best of our ability. Let me say that for lab on Thursday, I'll type this into the chat window. Okay, for Thursday's lab, please have some blank paper available and some colored pencils or pens, because you will be drawing during part of the lab activity. Okay, so if you don't have pencils or pens, please pick up a couple at the supermarket. You only need a few colors, maybe 3, 4, 5 colored pencils or pens would be helpful. Okay. All right. So any final questions before we get started with lecture? Okay. All right then, gang. Let's get into it here. Chapter 2. Last class, basically all I was able to do is show you a video talking about the anatomy of a cell. And I don't think we even took any notes on any of the anatomy of the cell. By and large, I'm going to expect that you already basically know the anatomy of a cell from your background in biology 101 and 102 and maybe other courses you've taken. But I will give just a couple preliminary notes to set the stage for a genetics course. Okay. So let's see what we can do. Okay. Chapter 2, and we'll take some notes on cell anatomy. Please, if you cannot read something, please let me know. If you have questions, please type into the chat window. Okay. All right. So let's begin here. Obviously, this is a genetics course, so we should probably mention the nucleus, right? I'm currently looking at figure 2.1 in your digital textbook. So if you have your textbook available, that's helpful. So figure 2.1 at this time. So you know that the nucleus is a membrane-bound structure. It's a membrane-bound structure that contains holes. We would typically refer to them as pores, that allows for the exit and entry of cargo into the nucleus itself. So things can go into the nucleus to modify the activity in the nucleus. Things can come out of the nucleus to modify activity in the cytoplasm. Okay, a membrane-bound organelle with pores that permit the exchange of material into and out of the nucleus. Okay, let's see what else here. So the nucleus, of course, is going to be the traditional location of the majority of the chromosomes you're going to find in a given cell. And again, I'll try to emphasize human cells in our conversation in this course. So the nucleus is the location of chromosomes. Okay. So let's see here. Let me ask a question in the chat window. Here we go. How many chromosomes are present in an N cell? N cell. How many chromosomes are present in an N cell? Have you come across this what manner of communicating before? How many chromosomes are present in an N cell? Okay, great. I'm getting some good responses for you. It's absolutely 23. Okay, so N is the haploid number of chromosomes. N is an arbitrary variable that the field of genetics has kind of just settled on and said every time we say N, we're referring to the haploid number of chromosomes in a typical cell. Okay, so the number of chromosomes in a haploid cell is arbitrarily referred to as the N of the cell. Okay, so a haploid sex cell, such as a sperm or egg, will have N equal to 23 individual chromosomes, right? Okay, and of course, most of the cells in our body are diploid, so that means they have a 2N status. Okay, most of the cells in the human body are 2N equal to 46 chromosomes. Okay, so the variable N refers to one complete set of chromosomes. One complete set of chromosomes. We appreciate that we are organisms whose cells are diploid. We get a full complement of chromosomes from our mom. We get a second full complement of chromosomes from our dad, right? So N refers to one complete set of chromosomes. And in our case, as a human species, is going to refer to the haploid cells, 23 individual chromosomes. Okay. Also, you appreciate that during the life of a cell, there's going to be many moments of time, many hours, many days, many weeks, potentially even many years. Well, the cells are not going through reproduction. That's not what the cell needs to do at that moment in time to support its job in the body, in our bodies. So during non-reproductive moments of time, the chromosomes don't really exist as chromosomes anymore. They exist in a much more dispersed state referred to as chromatin, right? During non-reproductive moments of time, which arguably is the majority of a cell's life, the genetic material is in a dispersed state known as chromatin. Okay, and so I don't want to hit this too hard, but certainly for this chapter, when we're thinking about the cell cycle, we're thinking about reproduction during mitosis and meiosis of the cell, we need to pay some attention to the organizational state of the genetic material. Okay, so, you know, for everyday common language, I'm just going to use the word chromosome, right? The cell has 46 chromosomes. But the fact of the matter is the chromosome may not really exist in a typical condensed state for most of its normal life. It kind of exists in a dispersed chromatin state. So if we ask the question, and I'll put this question in its own text cell in Mathematica, the question becomes, what is the difference between chromatin and chromosome, what would you say, folks? What is the difference between chromatin and chromosome? Please contribute in the chat window. If you'd really like to speak up, I'm happy to have you unmute your microphone and shout it out. So what's the difference between chromatin and chromosome? Okay. All right, so I see a response in the chat window from Jade and Natalie, and I appreciate that. Thank you very much. I'll just pause a little bit, give other people a chance to communicate if they wish. Okay. All right. So as we slowly continue here, you know, as Jade and Natalie have correctly indicated in the chat window, right? Chromatin and chromosome are made of the same molecules. Made of the same molecules, the same stuff is there. DNA is there, protein is there, and I dare say in the modern view of the chromosome or chromatin, RNA is there too. Okay, so the primary difference between chromatin and chromosome is its state of organization or lack of organization. It is the structure which is different. The material is the same, but the ordered arrangement of the material is different between chromatin and chromosome. Both chromosomes and chromatin possess the same building material, which is to say DNA, protein, and RNA, but chromosomes and chromatin organize the material differently. Okay. And as we've indicated in the chat window, as you've indicated, right? Chromatin is a loose, dispersed, and fairly randomly organized material. While chromosome is a much more tightly compacted, tighter, dense, and much more efficiently packed and organized material. Okay. So if I were to use a food analogy with you, I mean, it's lunch. Hopefully you've had lunch, so I'm not going to make you hungry by talking about this. But what I'm talking about, you know, if we were to think about a plate of pasta and some meatballs, a chromatin would be like somebody taking a spoonful of pasta and just slapping it randomly on your plate. The noodles are going everywhere, and there's a couple meatballs strewn throughout the plate, but there's no obvious organization to that material. You just kind of haphazardly dump it on the plate. That would be chromatin, okay? So analogy, haphazard arrangement of pasta and meatballs, okay? Whereas chromosome would be if somebody took the time to pick up a meatball, take one strand of pasta, and very carefully start wrapping a noodle of pasta around the meatball in a very tight, organized way, then that would be chromosome. Chromosome, you're going to organize the pasta, okay? Okay. All right, folks, please let me know if you have any questions. Otherwise, I will keep rolling because I kind of consider this background material that hopefully you have had at some other moment of your life in a biology course. Okay, so next we have the term nucleolus. Okay, the nucleolus is, you know, if you do have the luxury of looking at figure 2.1, you can see the nucleolus inside the nucleus. They're showing it there as in the picture as a darker staining pink region within the nucleus itself. The nucleolus is a region inside the nucleus that's responsible for manufacturing ribosomal RNA. Okay. Okay. Question in the chat window. What does ribosomal RNA do during the process of translation? What does ribosomal RNA do during the process of translation? The reason this is important for a genetics course is because if you express a gene, you're going to make a piece of RNA, and that piece of RNA then might get made into a polypeptide, or if I'm just a little bit sloppier in my language, a protein. So what does ribosomal RNA do during the process of translation? I mean, it does a number of things, but what's the thing that a biology 101 student should know and remember? Any thoughts? Any thoughts? Any guesses? Okay, maybe I got you on this one. Okay, so just to move the conversation forward, you might recall that ribosomal RNA is the structural component of the ribosome that forms the peptide bond between the amino acids of the polypeptide being made. Ribosomal RNA makes the peptide bonds between the amino acids. Okay. So I'll just put that down in your notes. Remember, ribosomal RNA makes the peptide bonds linking amino acids. It's very traditional that in a textbook picture, you'll get an image like you see in figure 2.1, where you see a nice nucleus and you see a nucleolus inside. Life is rarely that nice and neat. What I'm trying to tell you is that you can have multiple nucleoli inside an individual nucleus at one moment of time. You don't just have to have one. You could have 3 or 4 of them, depending on the cell, how large the cell is, and what its needs are for ribosome manufacturing capacity. How many ribosomes it needs? You need more ribosomal RNA, so you need to make more ribosomal RNA, so you have multiple nuclei building multiple ribosomal RNA. Okay, so a real cell can have multiple nucleoli inside the nucleus. Okay, next. Number 3, I'm going to refer to something that is kind of hinted at in figure 2.1, but isn't explicitly drawn there. I mean, kind of it is and it isn't. Let me explain. So if you look at figure 2.1 towards the bottom, you will see the term centriole. Well, the centriole is part of a larger organelle or region of the cytoplasm called the centrosome. Okay. So centrosome, let's give you some notes on the centrosome. The centrosome is an organelle of the cell that contains centrioles and a network of microtubules, as well as several other proteins which are still poorly defined, meaning that science doesn't have a firm grasp on this yet. It's an active area of research and characterization. Okay, so an organelle of the cell containing centrioles and a network of microtubules and other poorly defined proteins. Okay, that's just to be honest with you, right? There is a limit to our knowledge there's a limit to mankind's knowledge. There's some things we just don't understand, right? So it's important to admit that when we bump up against those walls. The centrosome, as I'm defining it here, is really the nucleation point. It's the point where the microtubules begin to assemble into larger structures, into tubes, okay? So the microtubules are long cylinders. Think about a garden hose. The garden hose could be the microtubule. But if you looked really close at the garden hose, you might realize that that garden hose is made of individual building blocks of materials that assemble together, that polymerize together to build the actual tube. Okay, so the centrosome is the nucleation point of microtubule assembly, which I can put the word polymerization in there. The microtubules polymerize, they form, they come together like Lego bricks locking into each other, well, within the physical geometry of the cell that we would call the centrosome. Okay, so polymerization. Okay. It's important that you appreciate that these microtubules that I'm referring to, they are in fact long cylinders made of interlocking proteins that are called alpha and beta tubulins. Alpha and beta tubulin proteins, which of course would be made by ribosomes, which of course have ribosomal RNA, which of course was made in the nucleolus of the cell. All of this stuff is connected, right? So microtubules are long cylinders made of polymerized alpha and beta tubulin proteins. Okay, so you take individual alpha proteins, individual beta tubulin proteins, you link them together like Lego bricks out of your toy chest. Hey, I like playing with Legos. Good stuff. A lot of fun to be had. You link these things together in just the right geometric pattern and you build the microtubules. Okay. All right. So the centrosome is visibly identified under the microscope by its two most prominent features, which are in fact the centrioles. So the centrioles that you see on figure 2.1 are in the centrosome. The centrioles are inside the centrosome. Okay. Centrioles exist inside the centrosome. And so at this point, I'm going to break out and give you a couple little talking points about the centrioles themselves, okay? And just to keep your notes kind of logically organized, I'll indent it here, okay? So the centrioles represent structures which themselves are made of microtubules. So the centrosome is the nucleation point of microtubules. The microtubules are made of alpha and beta tubulin proteins. If you take the microtubules and you arrange them in a concentric ring, you will build the centrioles, okay? So the centrioles are made of concentric rings of aligned microtubules. And the centrioles are typically found in pairs. You typically get a group of two in a typical non-dividing cell. Now, those centrioles will divide. They will, let me say it this way, those centrioles will reproduce. The cell will reproduce and double them when it has to go through mitosis or meiosis, okay? And we'll get into that in just a little bit. Okay. Centrioles are made of concentric rings of aligned microtubules. Typically, a single pair is found in non-reproducing cells. Okay. And some other little factoids for you. Okay, the centrioles participate in the function of larger important structures that use microtubules to accomplish goals. For example, centrioles are used to build the microtubules that form the part of cilia, right? So you think of cilia in your respiratory tract, right, along your bronchi and things like that that help move material out of the body. If you were to inhale some dirty air, right, you would cough it back up. The cilia is going to help move that material out so you can cough. All right. You're also going to have cilia in areas like your esophagus and things like that that help move material down into the stomach, right? So let's document that a little bit. In non-reproducing cells, centrioles help manufacture cilia. So think about the respiratory tract. They also help form flagella. Now, of course, in the human body, the only kind of cell that actually has a flagella is the male sperm cell. But if we think about microorganisms like bacteria and things like that, many of them will also manufacture flagella, and they have central-like machinery that achieves that goal. So flagella. And you may remember, if you've had any anatomy and physiology, that microtubules are a very important part of the cell's cytoskeleton. So this kind of cylindrical arrangement of girders and tubes in the interior of the cell that helps the main cell maintain its shape and overall structural integrity. And of course, it's not just like that, because there are machines that actually walk along the microtubules to move cargo back and forth in the interior volume of a cell. So the centrioles make microtubules that are part of the cytoskeleton. Okay, part of the cytoskeleton. Okay, I hope some of this is sounding somewhat familiar to you. Okay. In reproducing cells, which is kind of what we're going to focus on in our course, in reproducing cells, the centrioles will help manufacture and organize the spindle fibers that need to be present in order to attach to the chromosomes that need to get pulled around in different positions within the interior space of the nucleus so that the cell can copy them and divide them out equally and equitably between the cell that's going to divide. So you have the original cell and the daughter cell. So everybody gets, in the case of mitosis, an equal number of chromosomes. In reproducing cells, centrioles help manufacture and organize the spindle fibers, which attach to chromosomes to allow the cell to move chromosomes around. Okay, I literally mean to physically tug or push the chromosomes into different physical arrangements to allow cells to do what they in fact need to do. Okay, and so, okay, if you're very curious about, it's not a terribly magnificent picture, but if you look at figure 2.8, you can see the spindle fibers that are drawn in your textbook, figure 2.8. You see the spindle fibers that are attached to the kinetochore. The kinetochore itself is a multi-layered plate of proteins that act as an interface between the spindle fibers and then the actual genetic material, the DNA itself. Okay, so you'll be reading that in this chapter if you haven't already done so. That's figure 2.8. Okay, so I'll just highlight this as well, that during cell reproduction, the single pair of centrioles is duplicated. So you now have two pairs of centrioles in a cell that's getting ready to reproduce, okay? All right, folks, any questions right now? Okay. All right. These little teacher humor in the background here coming through the chat window. Please type yes if you are passed out right now. All right, good. I'm glad to see that you're actually still here. Okay, this is the reason we got to get back in the classrooms, right? Because I can't see you. I don't know what's going on. Okay, so let's continue. Any questions? All right. So the big deal about chapter 2 is really talking about the cell cycle, the process of nuclear reproduction and division in non-sex cells, which would be mitosis, and then the corresponding nuclear process of division in sex cells that create our gametes, our sperm and egg, which would be meiosis. Okay, so this is going to occupy our conversation today, Thursday, all of Thursday's lab, and maybe even a little bit next week. I don't know. It depends how this goes. Okay. So let's talk about the cell cycle. Okay. All right. So you have an example of a typical cell cycle in figure 2.5. Okay, figure 2.5 has a picture of a typical cell cycle. And we're going to sketch some things out. Excuse me, we're going to sketch some things out and do some drawing here in just a little bit. Well, let's put down some ideas here about what we mean by a cell cycle. So, you know, the cell cycle is kind of like the recipe for baking a cake. It's kind of like a recipe for baking a cake. You know, you get a bowl out of the cabinet, you put some flour in, you crack open an egg, maybe you put some milk in there, you agitate it around a little bit, you warm up the oven to 350 degrees, you shove it in the oven, you bake it a while, it inflates, it expands, right? All this kinds of stuff. The cell cycle is like a recipe, not for baking a cake, but for living life and making a copy of the cell. All right. So let me define it maybe a little bit more cleanly here. The cell cycle should be thought of, or at least can be thought of, as a recurring or cyclical series of molecular events that transition the cell between proliferation, which is to say reproduction, which is to say copying itself, and non-proliferation, which is to say not reproducing and simply kind of living the normal humdrum boring life that cells live. Okay, and key in the idea of the cell cycle is in fact that this is a cyclical process, or at least it can be. It isn't always. It can be, but it isn't always. Okay. It is possible for cells to go through part of the cell cycle and kind of just stop for a whole variety of reasons. Okay, so sometimes that happens. So what I'd like to do, and what I want to encourage you to do also, is doing some drawing at the house. Okay, so I much prefer to be in class with you on the whiteboard. This just goes better. And we'll do that next week. But at this time, what I'm going to do is vaguely reproduce figure 2.5 in your notes here and talk about some features of things along the way. Okay. One more note I'll make here before I do my drawing, and that is that, in human cells, which obviously are eukaryotic cells, the cell cycle comes in basically two varieties, okay? And what I mean by this is probably what you're guessing at. We have somatic cell cycle, which, when I say somatic, I'm talking about a non-gamete forming body cell, okay? Somatic, or a non-sex cell, okay? The cell cycle in that is going to involve a period of activity known as interphase, plus a period of activity known as mitosis. And we're going to talk about these things in considerable detail. The alternative is that you're going to have a germline cell, a sex cell stem cell that's tasked with the responsibility of forming the haploid sperm and the haploid egg in males and females respectively. And you're going to have interphase plus meiosis, okay? So there is a lot of important commonalities between somatic cell cycle and germline cell cycle. And there's also some significant differences that we're going to point out along the way. Okay, so let's talk about the somatic cell cycle. The somatic cell cycle. So that's the non-sex one. That's the one that's going to involve mitosis. Let's talk about this a little bit. And so hold on a second here. Yeah, okay, not what I wanted. Let's go this way. Okay, here we go, folks. So, all right, so let's make, and encourage you to do your own drawing at the house. Let's make a nice circle here. I'm sure yours will look nicer than mine. Okay, so this is the typical circle of a cell cycle. And the first point of it is to appreciate that a circle really has no beginning or end, does it? It's a geometric object that really has no beginning or end. You could point to a position on the circle and go clockwise or counterclockwise around it, and hey, you're going to wrap around and start again, right? That's the whole point of this being a cyclical process. Okay, so we have the typical cell cycle here. There's some features of this that I would highlight for you, and I'm just going to change my color that I'm using with you. Let's go with green, I guess. It doesn't really matter, just as long as it kind of sticks out a little bit. And we'll go along the interior here. Okay, so everything inside the green is in fact interphase. Come on, technology. Oh, sorry. This should be working better. I don't know why it's not working. Come on. Oh, this is why we need to get back to the classroom. Oh, my digital pen does not want to draw for some reason. Oh, this is irritating. Come on. I'm going to really be stuck if I can't get this to draw. Oh, good grief. No, this is not very good at all. Okay, so backup plan number one. Let me go get a cell cycle image from the internet. Okay, hold on, folks. Sorry about this. This is not what I wanted to have happen. Okay, maybe we can use something like this. And I'll try this. Okay, so we have a rough cell cycle here, okay, that you see on the screen. And I doubt my digital pen's going to work here, but you appreciate that interphase is basically G1, S, and G2. G1, S, and G2 would be interphase here. Let's see, is this going to cooperate at all? Why is it working now? Okay, all right. So G1S and G2 is interphase. Let's take some notes here. Interphase equals G1, S, and G2. And if I'm being completely honest, it also includes G0. Okay, although G0 is often thought of as cell cycle arrest, meaning you're leaving the cell cycle and kind of going into a hibernating state. Okay, that typically happens at some moment of G1 where you kind of go out of the cell cycle. Okay, so let's talk about G0 and then we'll come back in and talk about the rest of things. Okay, so G0 equals cell cycle arrest. Okay. This typically happens at some moment during G1, okay? And with this kind of scenario here, all right, let's make this talking point 1, talking point 2. The cell basically goes into a state that's called senescence, okay? G0 puts the cell in a senescent state, okay? There we go. All right, so if we think about cell cycle arrest or senescence, you know, you might think that that's a bad thing, but it's not always a bad thing. This is a very contextual thing, okay? So let me say this. Sometimes cell cycle arrest is good, and other times it is bad. And so let's think about this, okay? You would want a cell to stop reproducing if production of that cell would cause harm, right? So in that case, cell cycle arrest would be a good thing. And so scientists believe that cell cycle arrest, the so-called senescent state, is one protective mechanism that Mother Nature has created, has evolved to try to suppress tumor formation and subsequent cancer development, okay? So cell cycle arrest, so let's do it this way. Good aspects of cell cycle arrest. Okay, preventing tumor formation or cancer development, okay? We don't want those cells to continue to grow and reproduce because they might end up overtaking the body and leading to the death of the organism, the entire human, right? There can be other times, right? During embryonic development, before you're even born, you want to be sure that your internal organs are growing, reproducing, and developing at the proper rate, but not at the incorrect rate. What I'm saying to you is your heart, the primitive embryonic heart, gets to a certain size and you don't want it to overgrow, because if it overgrows too fast, it will outgrow the cavity within the body that it's forming, and that could lead to the death of the embryo, right? So embryonic development, there are going to be times when you purposefully want cell cycle arrest so the internal organs grow to their proper sizes and shapes and dimensions and don't overgrow, because overgrowth would ultimately probably lead to a miscarriage. Okay. Okay. So these are two really good aspects of cell cycle arrest. We prevent cancer and we ensure that our organs develop properly. There's another aspect which I think we could probably say is good, if not maybe perhaps neutral, and that would be the following. Okay. Some tissues and organs of our body, you know, if you've had AMP, you know, some cells, once they're created and they grow to their proper dimensions and sizes, well, they stop reproducing for most of your natural life. You know, neurons would be one of them, right? Neurons don't tend to reproduce unless there's some very special circumstances that happen. And of course, we know folks that unfortunately have had brain damage through strokes, and sometimes the person never recovers. Okay. But so what I'm saying is there are organs that live their natural life out in an arrest with cells that are in an arrested state. And as long as there's not trauma that damages the brain, as long as there's not trauma that damages the liver, then everything is okay, because that's how Mother Nature wanted our bodies to work. Okay, so let's put it this way: a neutral aspect of cell cycle arrest is the fact that certain organs, certain tissues, certain cells basically they live out their life in a cell cycle arrest state. Okay, and so I'm talking about neurons of the brain. They don't tend to reproduce. I'm talking about liver cells. You know, liver cells will reproduce if there's liver damage, if there's a physical laceration or something like that, they will reproduce, but generally they don't reproduce in your life. And I'll stop with this last one, skeletal muscle, right? Skeletal muscle cells don't generally reproduce. They can get larger because you're increasing the number of myofibrils in them when you do weightlifting exercises, but you're not stimulating and reproducing muscle fibers. Okay, so that's a neutral aspect of cell cycle arrest. I guess you could take the neutral aspect and say, well, that becomes a bad aspect if there's damage to the body. If you're in a car accident, something like that, then it's a bad thing that the brain won't make new cells to heal itself, right? That's a bad thing. Okay. And then we have arguably a negative aspect of cell cycle arrest. And this is where we come up with the idea of aging. Okay, as we get older, as we go from 20 to 30 to 40 to 50 to 60 and so forth, as we get older, most of the cells of our body, an increasing percentage of our cells start to develop, they start to adopt a cell cycle arrest phenotype, a senescent phenotype. And that senescent phenotype seems to increasingly, with age, correlate with inflammatory processes that are still poorly understood, but seem to be characteristic of the aging process. What I'm saying is that your body when you're 50 is certainly not the same kind of body you had when you're 20. Okay, so as cells age, they increasingly adopt a cell cycle arrested phenotype. Aging correlates with inflammation. And of course, inflammation correlates with negative outcomes for the quality and length of your life. Okay, this is an active area of research in the research community. There are a lot of companies out there that would like to find therapeutic agents which can slow down the aging process, because there's money to be made in the drug fountain of youth, so to speak. But long story short, if we could find some of the molecular mechanisms underlying cell cycle arrest in aging cells, we might be able to improve quality of life in 60, 70, 80, 90-year-old folks. Okay, so there is a lot of research out there, companies working on this. Okay, any questions so far? Okay, let's talk about the interphase business. Okay, and so probably it makes more sense for me to reorganize my notes here. Sorry about that, but I'm going to do that just to make things a little bit better when you actually get these. Okay, let's talk a little bit more about interphase G1S and G2. So I'll make this bullet point number 2. Let's see, this stuff didn't work out for me, so I'm going to get rid of it. Okay. All right. So let's continue here. So interphase is the time in between reproductive events in the nucleus. The time in between reproductive events in the nucleus. Okay. It is characterized by this G1S and G2 phases. So let's document this. G1 stands for growth phase 1 or gap phase 1. And some of the major properties of the first growth phase include the following characteristics. Okay, during growth phase 1 or gap phase 1, you're going to have many of the cells organelles double. They reproduce. Okay, so you're going to make more mitochondria. You're going to make more endoplasmic reticulum. You're going to make more vacuoles. You're going to make more ribosomes. All of these things are going to double in number, because obviously when the cell finally divides, the new cell that's being made is going to need copies of all the functional machinery to do the normal living of a cell. Okay, so let's see here. Organelles double, so which is to say they are reproduced. So that a new cell can eventually have correct machinery to live. Okay, and this is especially true of the mitochondria for production of energy and the ribosomes for production of protein, because without the protein, you don't have the enzymes, you can't support the chemistry of life. Okay. It is in the it's basically in G1 that the centrioles that we documented just a few minutes ago will also double in number. Sometime between the transition of G1 into the S phase, the centrioles are going to divide. Okay. Let's see here. Please stop me along the way with questions if you have them. Okay, otherwise I'm going to keep rolling because there is so much content in this chapter. The S phase. Okay, the S phase stands for synthesis phase. Okay, synthesis phase. And the big deal here is that the DNA of what we think of as the chromosomes is in fact going to reproduce. Okay, it's going to reproduce. So DNA is copied. All right. So question in the chat window. Here we go. How many nucleotide pairs of DNA are found in a typical non-sex cell that is not reproducing? How many nucleotide pairs of DNA are found in a typical non-sex cell that is not reproducing? Folks, I just asked you, whether you appreciate it or not, I just asked you, what is the size of the diploid human genome? Okay, just to move things along, and if you're trying to find the answer, thank you for doing that. I appreciate that. My point here is I'll make this point. Okay, so I'll go back up under G1 and I'll highlight that in G1, the diploid human cell, because we are talking about mitosis. So this is a non-sex cell. We're talking about the cell cycle, which includes mitosis. Typical cell is going to have about 6.2 billion nucleotide pairs of DNA. Okay, this is the diploid. This is the size of the diploid human cell. Okay, this is the size of the diploid human genome. 6.2 billion nucleotide pairs of DNA. Okay, and so just type into the chat window for me. After the S phase has completed its work, how many nucleotide pairs of DNA are going to be present? Once again, after the S phase has finished its work, how many nucleotide pairs of DNA are going to be present? Okay, thanks, Connor. And I'm just going to run with, okay, thanks, everybody. Appreciate it. Okay, 12.4 billion. That's right. So in an S phase cell that has successfully copied the genome, you're going to have roughly 12.4 billion nucleotide pairs of DNA. Holy cow, that's a lot, right? Once DNA is copied, the non-sex cell will have roughly, and again, this is roughly, 12.4 billion nucleotide pairs of DNA. You think about that, right? Think about having 6 pairs of socks in your dresser drawer, and all of a sudden they become 12 pairs of socks. Okay, this is a significant thing, tremendous outlay of energy to manufacture that DNA. Okay, let's continue here. Next, we have G2 phase. Okay, G2 phase is gap phase 2 or growth phase 2. Okay. And so, gap phase 2 includes activities that are considered quality control activities to ensure that the cell has successfully achieved what it should have achieved during growth phase 1 and S phase. So gap phase is going to make sure that we really did get a proper copying of the DNA. Gap phase 2 is going to ensure that we really did get proper copying of the centrioles in G1. So think of G2 as a quality control phase where we make sure all the necessary components to manufacture a new cell have successfully completed themselves and we're ready to begin reproduction. Okay. Quality control phase where the cell attempts to measure if key events like DNA doubling and organelle doubling have succeeded so that mitosis can begin. Okay, so all right. So this leads me to a couple things to talk about here. I don't know. I'm not sure if my digital technology here is going to work with me. It's very frustrating, but One of the things we're going to focus on, or really I should say you are going to focus on in your lab on Thursday, is the idea of cell cycle checkpoints. And so let me just put a couple things in here, if I can. I don't know if it'll work or not, but let me try putting a couple ideas here. Hold on a sec, got to look at my drawing here. Okay, so I'm going to put, I hope, a triangle here. And just put the letters C, CP for checkpoint. So there's a checkpoint between G1 and S that I'm going to document for you. So here we are in G1, going to going towards S, and there's a cell cycle checkpoint in here. There is also a checkpoint between G2 and mitosis. There's actually many checkpoints that could be talked about. We won't talk about all of them, and I'll be honest, I doubt very much science even knows of all the checkpoints that exist. Okay, so there's going to be a checkpoint here. Sorry for the really poor drawings. And then there is a checkpoint in part of mitosis, which is known as prometaphase. Okay, so you know that mitosis, if you've read ahead, you know mitosis is made of prophase, metaphase, anaphase, and telophase. So roughly in the middle of prophase, there is an additional checkpoint. So I'm just going to put a second triangle here to fit it in here. And I will document these things in your notes. Doggone it. Sorry, it's so ugly. My digital pen is not working very well. Okay, so let's talk about cell cycle checkpoints. And as I said, you will get more of this in lab on Thursday. Okay, cell cycle checkpoints. Okay, so once again, this graphic that you see on the computer screen is showing you a circle. This is the cell cycle. If we have G1, S, and G2, if the cell is actively engaged in metabolic activities that are characterized by humans as G1, S, or G2, then the cell is in that period we call interphase between reproductive processes in the nucleus. And then once you get out of G2, you go into mitosis, and we'll talk about that, well, probably mostly next time since I'm running out of time here, but let's see what we can do. So let's talk about the G1 to S checkpoint. G1 to S checkpoint. So for the G1 to S checkpoint, really just what I want to tell you is the whole purpose of the G1 to S checkpoint. So in the cell, there's going to be machinery. And when you're talking machinery, you're talking about a lot of proteins, a lot of enzymes. There's machinery, there are molecules that are going to evaluate the integrity of the DNA. In other words, is the genome ready to be copied? Is it ready to be copied? Okay. Do we have too few chromosomes? Has something happened that we're missing chromosomes? Has something happened that we have an unusual number of chromosomes to start with? For some reason, do we have more chromosomes to start with than we're supposed to have? Well, the G1 to S checkpoint is going to try to evaluate the answer to those questions. Okay, so the machinery of the cell will attempt to measure the integrity of the genome. Do we have the proper number of chromosomes present? Are there too few or are there too many? If we don't have the proper number of chromosomes present, then it doesn't make any sense to go into the S phase, because we're going to make the wrong number of chromosomes in that process. And that's probably not a good outcome. We're probably on our way to something like a tumor. Okay, so stay with me. Let me finish these ideas, and I promise I'll let you go. B, the G2 to M checkpoint. So that would be the second, that would be this checkpoint up here, the G2 to M checkpoint. The G2 to M checkpoint okay. The purpose of this checkpoint is to basically ask the question, okay, we've just been through the S phase, and I'm trying to be sure that everything looks good so I can start the nuclear process of separating the cell into two equal cells. So the G2 to M checkpoint is going to use its machinery to determine if the DNA has in fact been properly and successfully copied. Okay, and I'm racing here a little bit, but the third checkpoint that I wanted to talk to you about is the checkpoint that's inside mitosis. So this little green square here that's overlapping with the mitosis diagram. Okay, the M or mitosis checkpoint, and this is really something that's in pro-metaphase. The mitosis or pro-metaphase checkpoint. Okay, its job is to evaluate whether the spindle fibers have successfully attached to the kinetochore, which is attached to the chromosomes. Because if it hasn't, then the cell will not have the ability to move the chromosomes in an equitable and evenly divided fashion between the cell, the father cell, and the daughter cell, between the mother cell and the daughter cell, between the original cell and the cell that's about to be manufactured. Okay. So the machinery will attempt to determine if spindle fibers have successfully been attached to the kinetochore of the chromosomes. Okay, because again, if it hasn't, then we're going to end up with two cells at the end that have an uneven and unequal distribution of chromosomes. Once again, that is a recipe for cell death or something worse like organismal death through tumor and cancer formation. Okay, folks, I know I'm 3 minutes over time, so I'm going to stop, I promise. Okay, lots to do. Any questions right now? Okay, so for Thursday, please try to have paper and colored pens or pencils for lab. And Thursday is going to be cell cycle, mitosis, and meiosis day. It's going to be coming out your ears, folks, because we're going to really hit it hard, okay? Have a great afternoon, and I'll see you Thursday.
Thank you.
Bye-bye.
I did have a question.
Okay, Anthony.
When you were talking